is Patrick Giraudeau. I am a professor of analytical chemistry at Nantes University in the west of France. And I am leading a team of approximately 30 people that involves several PIs in NMR, students and technical staff, and also leading an NMR facility here. And I'm part of the CESAM Research Institute, which is a molecular chemistry research institute at the University of Nantes. So our main research field initially is to develop new methods for the analysis of complex mixtures based on NMR, and more specifically, quantitative methods. So what we want to do is to be able, in complex chemical and biological mixtures, to quantify multiple analytes at the same time. And for that, we like to design new experiments, what we call in NMR pulse sequences, which are different ways of playing with nuclei in order to get the answer that you want to get. And then these experiments, we implement them on a variety of spectrometer, high field spectrometers, but also on benchtop spectrometers. We have also a very original development project, including the development of hyperpolarized methods in collaboration with Bruker, actually, through a prototype system that was installed a few years ago. We've been recently achieving very interesting results in different fields. I could mention maybe two of them. The first one is the development of new ultra-fast 2D NMR methods, which are specifically tailored for the analysis of samples that evolve in time or that need to be analyzed in a very short time. And the second one is first applications of hyperpolarization, and in particular, a technique called dissolution dynamic nuclear polarization to metabolomics. So this is a technique that is not new, that we did not invent, but I think we are among the first to investigate the potential of this technique for the application to metabolomics and to complex mixtures. And this one is something we achieved in collaboration with Bruker, and in particular with Jim Kempf and his team, who provided a prototype of dissolution DNP equipment dedicated to analytical chemistry. And that these results are achieved through two ongoing ERC projects in the team, one which is led by myself, and the other one being led by Jean-Nicolas Smith, another PI in the group. So I believe that the research we are doing has some kind of societal impact, maybe not directly, but we are developing new methods, for instance, for metabolomics, that in the end will hopefully be helpful for clinicians, for instance, to design new personalized medicine solutions, to better diagnose some specific pathologies. So these are applications in health. In the field of food science, it can help to get simply better food, or to design better plant varieties. The applications are extremely diverse. There are also some applications of the quantitative methods that we develop in the pharmaceutical industries to better control the quality of drugs, for instance. And more generally, what we try to provide is to provide solutions that will help people working in more applied fields to get a better understanding and a more accurate understanding of the chemistry of life. In terms of perspective, obviously, we're going to spend a lot of energy in the next 10 years to increase even further the performance of NMR, which still has some limitations for the applications of complex samples. So we're going to work on solutions to make it more sensitive, relying on our proof of concept work on hyperpolarization that obviously needs to be taken to the next step. We also want to continue working on making NMR spectroscopy better resolved by providing the experiment that can better separate overlapped resonances, for instance. And making NMR spectroscopy more accessible to the real world, to clinics, for instance, is also something we have in mind. And that will obviously rely, at least in part, on more accessible equipment such as benchtop spectrometers.